Okay, so last week we talked about deterministic models in epidemiology, how we can use deterministic differential equation models to track the spread and transmission dynamics of different infectious diseases. And the thing about these models is that they were all deterministic. So given a given set of starting conditions and a given set of parameters, there's only one possible outcome that could play out in these deterministic models. This week, we'll move on to consider stochastic models that can be used both to model epidemiological dynamics, as well as in molecular epidemiology to, for example, simulate phylogenetic trees and sequence data. And then next week, we'll, of course, try and put everything back together using phylogenetic models that try to combine the epidemic models that we've been talking about in these past 2 weeks with phylogenetics. Okay, so first off, what do we mean when we talk about stochasticity? Well, we've already talked about stochastic models or stochastic processes at a few different points in this class. We talked about a stochastic process as basically being a set of random experiments playing out over time. So if we start a stochastic system from a particular initial condition, the randomness in the system will lead to different outcomes over time, unlike what we would expect in a deterministic system. But why do we need to include stochasticity in our models? There's 3 main reasons, at least that I can think of, for why we need to include stochasticity. The first is to actually include true randomness in our models. We can think of some processes as being truly random. And then the second reason is that maybe the process that we're considering, or the model that what we want to model, isn't actually truly random, but there's some process that we're uncertain about, or it contains a lot of heterogeneity. And so we can use stochasticity as a way of taking into account our uncertainty about that process. And then the third reason why we might want to consider stochasticity is, in a statistical sense, we might want to include stochasticity in our models to capture variability that we observe in real-world data. So let's go through these one by one. So as I said, there's some processes, for example, like rolling a dice, or the behavior of subatomic particles in like electrons in quantum theory, that we can think of as behaving truly randomly or stochastically. But it's debatable, sort of both scientifically and philosophically, whether or not any system truly behaves randomly or stochastically, or whether there might actually be an underlying deterministic process that we could use to explain the behavior of the system. We just might not know enough about that underlying process that's giving rise to what we're calling stochastic dynamics. And I think this is what Einstein meant when he made this famous remark about the nature of quantum dynamics, when he said that I am convinced that God does not in fact play dice. What he meant by this was that we may think of certain processes as being random or stochastic, but in his worldview at least, he always thought that there was some underlying deterministic process, that if we just knew enough about how the world really works, we could describe real-world phenomena in terms of these deterministic models. So generally, it's the case that rather than thinking about processes as being truly random or stochastic, it's just more convenient to think about the outcome of certain experiments as random, or the outcome of a particular process as random. Think for a second about the physics of rolling a dice. It's very easy to model this as a random process. We could just say, that the probability of rolling any number on a 6-sided dice is just equal to 1/6. It's fairly easy to model that as a random process. But if we wanted to model the same process deterministically, we could, but we would have to know a lot about the physics of dice. And we'd have to know a lot about the initial conditions when we first sort of tossed the dice into the air. And we'd have to know a lot about the angular momentums and the velocity of the device as it traveled through the air. And we'd have to know things about the environment too, right? We'd have to know things about wind speeds or resistance in the air that the dice encountered before it landed. So it'd be very hard to model the throw of a dice using deterministic equations, even if in fact we do know enough about the physics of how the physical world works to actually be able to do that. So what happens then is, rather than thinking about the world as being truly random or stochastic, we generally think about processes at either much smaller scales or much larger scales than we're really interested in modeling as phenomenological models. So I'll pause here and just ask you all, do you know what phenomenological models are? Can anybody give a practical definition of phenomenological models?
Maybe like an SIR model where the rates are based on what's happening.
So what exactly in an SIR model would be phenomenological?
The fact that susceptible individuals become infected become recovered.
Yeah, I think you're getting at the heart of it, right? So we treat it as sort of a matter of fact that when a person becomes infected, they'll eventually recover, when in reality, there's a bunch of within-host processes, including immune responses, that determine whether an individual host actually recovers from infection. And that's the sense in which I mean phenomenological here, just the sense that we're modeling something, not using any sort of underlying theory about the mechanisms of how it works. We're not using any sort of mechanistic model to describe how the world around us works. We're just treating it as if it happens. And that's what we mean by phenomenological here. To give another example from sort of the epidemiological world, we can think about transmission rates between different, say, plant hosts in a field over time. We might treat transmission rates between different hosts as if they were stochastic, but there might actually be deterministic underlying reasons why the transmission rate between different plants in a field varies. It might vary due to the fact that different leaves on individual plants have different numbers of spores, and these individual spores might vary in terms of their viability. And there also might be some sort of variation or heterogeneity in terms of environmental conditions that affects whether a given plant is able to sort of transmit to another plant through time. So another reason why we use stochastic models is to take into account our own uncertainty about the microstates of these systems, that we really just don't know enough about the states of individual hosts and what's going on inside them to be able to sort of model model heterogeneity in individual outcomes. And so this is another reason we use stochastic models as well. In this sense, stochastic models are sort of acting as a bet hedging strategy, where we're hedging against our own uncertainty about what's going on at these smaller scales than we're really interested in modeling. Okay, and then the third reason why we might be interested in using stochastic models is to try and capture the heterogeneity and variability that we see in real-world data that we often can't capture with our simple deterministic models. And this is important both for doing statistical inference when we're trying to fit a particular model to real-world data, because if we just fit a deterministic model to real-world data, our model might be too rigid and not sort of allow for the variation in the processes that actually produced the data. And of course, when we're actually simulating data that we might want to use to, say, test a particular method, we also want to include some form of stochasticity to allow for different possible outcomes. If we just Simulated on a deterministic model, we would get the same outcome over and over again, and that wouldn't be very interesting. But simulating under a stochastic model allows for us to get an ensemble of different outcomes that we can then use to test our methods. Okay, so to review this part of the lecture, I just wanted to say that there are maybe a few processes in nature that we can think of as being truly random, although as we saw in the case of rolling a dice, maybe those stochastic processes are actually deterministic at some level. So generally, when we include stochasticity in our models, it isn't necessarily to capture fundamental randomness in a given system, but it's rather to take into account our uncertainty at what's going on at sort of levels far below or far above, in what we're actually modeling, or to capture variability in data in a statistical sense. Okay, so that was why it's important to include stochasticity, but now we have to consider how do we include stochasticity in our system. And how we include stochasticity really depends on the type of stochasticity that we want to consider. And we can really think about two different main types of stochasticity. There's what's known as observational noise, and then there's what's known as process noise. So observational noise is just errors in our observations or due to our limits in the level at which we're able to observe a particular system. The classic example of this in infectious disease epidemiology would be that there's just oftentimes misreporting in case reports or the number of infections over time. So there might be underreporting or overreporting, and that would be a form of observational noise. But in a sense, in terms about actually modeling dynamical systems like infectious disease dynamics, observational noise isn't actually that interesting because it doesn't actually affect the system under study. It doesn't change the future dynamics of the system. It only changes our observations, which might be important to consider when we're modeling the data statistically, but it's not as exciting to consider when we're modeling the dynamics of these systems. So the second main type of stochasticity that we need to consider is what's known as process noise. And this is really randomness in the processes that are actually driving the dynamics of a system. So randomness, say, in the infectious process or the infection process in an SIR model. And in turn, we can sort of divide process noise into two different subtypes. There's what's known as environmental noise, and then there's what's known as demographic stochasticity. And both of these are sort of rather unfortunately named because environmental noise doesn't actually have to be environmental, and demographic noise doesn't actually have to be demographic, but they're just the terms that are widely used, at least in the population dynamic and epidemiological modeling literature. And so what we're going to use these terms operationally. And so when I say environmental noise, what I really mean is that there's some internal or external perturbations changing a particular parameter or the rate at which a particular process is happening in our system. So a good example of environmental noise would be sort of climatic fluctuations in a variable, say like relative humidity or temperature, that might be affecting the transmission rates of a respiratory pathogen or a foliar pathogen on a plant. But it doesn't necessarily have to be a climatic factor. It could also be something internal to the behavior of individual hosts as well. It could be, for example, that there's random fluctuations in the transmission rates of an SIR model because different numbers of people are out traveling about and contacting one another on a given day at a particular time. So we'll refer to this all as environmental noise. And then the second form of stochasticity, and the really interesting form of stochasticity that we'll spend a lot of time talking about today, is what's known as demographic stochasticity. And this is randomness in the timing and the outcome of individual events. When we actually think about individuals in a population, there's going to be some randomness in terms of the fates of these individuals. Individuals are, say, going to be born or die at different times, or a given individual is going to become infected with a disease or recover at different times. And so it's this randomness at this individual level that we're really interested in these models, because ultimately, fundamentally, at the end of the day, unlike in the types of deterministic differential equation models we were considering last week, in the real world, we have to consider whole individuals. We can't have fractions of individuals get infected. We need whole integer numbered a number of individuals in our system. And so that's why we'll spend a lot of time talking about demographic stochasticity. Just to go back to environmental noise just for a second though, it's relatively easy to include environmental noise in our models. A classic example from statistical models is just to include some sort of form of random noise term in our model. Say, for example, we had a classic regression model where we were modeling some outcome y as being the product of some explanatory variable x times a regression coefficient beta. We could add environmental noise into that system just by adding an air term, this epsilon term in this equation, which accounts for the fact that there might be some external noise seeping into the system. There might be factors that we're not modeling that cause the relationship between x and y to vary slightly from what we would expect under a strictly linear relationship. So we can include noise in these types of models just by adding some sort of random noise term to the model. And we can do the same things for dynamical systems, including SIR type models. We can add noise to the rate at which particular rates occur in our differential equation models using what are known as stochastic differential equations. And we're not going to go very deep into these at all here. I just want to point out that it's a relatively easy thing you could do if you wanted to. For example, if you're interested in adding environmental noise into our differential equation for the number of infected individuals through time, we could have our standard differential equation of beta Si minus gamma I, so transmission minus the recovery rates. But then we could add a third term to this equation that accounts for randomness in the rate at which transmission events are actually occurring. And so this chi term, this weird Greek letter here is chi, could be thought of as a random normal variate, like we introduced in our regression equation above, that just allows for a little bit of noise or randomness to enter into the process. So if that noise term was below 0, we would have slightly fewer transmissions than we would expect deterministically. And if it was above 0, then we might have, if it was below 1, we might have slightly less infections, and if it was above 1, we would have slightly more infections then. And so that's how we can include environmental noise in our classic differential equations. But as I already alluded to, what we're primarily going to be interested in is demographic stochasticity, because this is the form of stochasticity that we need to add into our models to be able to model the world in a way that we actually think that the world works in terms of having a discrete number of individuals, and the type of stochasticity that we'll be really interested in adding to our simulations that will allow us to simulate phylogenetic trees and sequence data. So fundamentally, what we have to consider when we start thinking about demographic stochasticity is that there's randomness in the outcome of particular events like transmission events or recovery events. And this randomness arises because we're treating individuals as discrete whole units. So there always has to be an integer number of individuals in our population. We can't have half of an individual. We have to have whole numbers of individuals. And so when we start thinking about these individual level processes, it starts to make sense to think about individual events as reactions, kind of like we think about reactions in chemistry. So in chemistry, if two different molecules react with one another in a chemical reaction, there has to be a whole number of molecules there. We have to have, say, a whole molecule of water interact with a whole molecule of carbon dioxide, just to give a chemical example. We can't have fractional values for the number of individual molecules in a reaction. And we'll start thinking about processes happening in a population of individuals in the same way as reactions occurring amongst whole numbers of individuals. So we can think about our SIR model, our base SIR model without any host demography from last week in terms of a stochastic SIR model with demographic stochasticity. And so really there's two types of events or two types of reactions that can occur in such a model. There's transmission events. So two hosts, a susceptible host and an infected host, can come into contact with one another with some rate beta. And this reaction will give rise to two infected individuals, or 2I in this equation here. And likewise, we also have to think about recovery events, which converts infected individuals into recovered individuals who can no longer be infected, which we'll assume happens at some rate gamma. So how would we actually simulate under a model like this with demographic stochasticity? Well, the interesting thing that we have to consider here is now that we have many different individuals in our population and different types of individuals, say susceptible, infected, and recovered individuals, and these different individuals can be involved in different types of events, transmission events or recovered events. So what we have then is a system where there's multiple competing processes. There's sort of dueling processes going on in the background. We can either have the next event in our population be a transmission event, or it can be a recovery event. So in order to simulate under such a model, we have to have a way of deciding when the next event occurs and then what type of event occurs at the next time step. So there's a very simple way of doing this, and it's basically a three-step process that gives rise to what's known as the stochastic simulation algorithm, which is very generally used across basically all of the sciences, both the physical sciences and the life sciences. And so it's a three-step process. In the first step, we'll go through these one by one. In the first step, we have to simulate or draw the time to which the next event occurs, at which the next event occurs. So step one is finding the next event time. And we can break this one up into three sort of sub-steps or individual things that we have to do in order to find the time of the next event. So the first thing we have to do, step A here, is we have to first compute the rate at which each reaction or event type occurs. And so again, in our SIR model, we have transmission events and we have recovery events. And so transmission events, we're going to say occur at a rate lambda t, and then this is equal to our incidence rate, beta SI, that we've seen over and over again by now. And then we have recovery events, which we'll say that those happen at rate lambda r, and that's equal to gamma times I, the number of infected individuals times the recovery rate. So then to determine the time at which the next event occurs, we can sum these individual rates together to give us the total rate at which all events or all reactions are happening in our population. And we'll refer to this as the total rate or lambda total. And then all we have to do in order to draw the time to the next event is just to draw a time from an exponential distribution with a rate parameter lambda total. So if you recall, we've seen the exponential distribution at least twice before in this class. And the exponential distribution describes the waiting time to when any event occurs, given that that event occurs at a constant rate. So for example, we encountered the exponential distribution when we were talking about the waiting time to the next coalescent event. And we saw that the rate, or the time to the next coalescent event is distributed exponentially. The same thing here. When we think about the time to the next event that occurs in our model or in our system, that waiting time will be distributed exponentially. So we can just draw an exponential random variable from an exponential distribution to get the time to the next event. Okay, so we know when the next event is going to occur. The second thing we have to do then is to choose what type of event occurs. So in our simple SIR type model, there's only two types of events, transmission events and recovery events. So what we can do is find the relative probability of either event type happening. And so when we have dueling processes like this, we have sort of two events that can both happen. The way that we pick which one happens first is we compute the relative probability that each event occurs first. And the way that we do that is just take the rate at which a given event occurs, say transmission rates, and divide it by the total rate at which all the other events occur, the sum of all the other rates. And so by doing this, we can find the relative probability of a transmission event or a recovery event being the next event. And so then we can just choose what type of event occurs at the next step according to these relative probabilities, which is analogous to taking a single draw from a multinomial distribution. Okay, and so now we know when the next event happens and we know what type of event happens next. So we can now update our system to reflect the occurrence of that event. So say that the next event is a transmission event, we can update the system accordingly. So we would have to do three things here. We would have to update the time, our current simulation time. So if we were at time t, and the next event occurred at time t plus delta t, we would just reset our time variable to be that next event time. And then we have to update our state variables, which is here, the number of susceptible and infected individuals. So if a transmission event occurred, we would decrease the number of susceptibles by 1, and we would increment the number of infected individuals by 1. And that's basically our three-step process through which we can simulate the stochastic dynamics of any system with demographic stochasticity. The only thing that we would actually have to do to turn this into a real algorithm that we could run would be that we would have to iterate this process repeatedly of choosing the next event time, picking what event happens, and then updating our system. And we can do this repeatedly until we reach some end condition, at which time we have stopped. And the most likely or the most usual sort of use case is that we would iteratively, sorry, coughs are always worrying these days, but I think that was just, I have something in my throat cough, sorry. Yeah, so the time to the next, or end condition could be something as simple as you just simulate the dynamics to a particular stopping point sometime in the future. Or we could set a particular end condition, say like having the disease completely die out from the population, at which time point we would stop our simulation. So we can simulate the dynamics of an epidemic under this type of model. And what we see is that there'll be variation or differences in the outcomes of our simulations. So in some epidemics, the outbreak will take off and the number of infecteds will rapidly increase through time. Whereas in other epidemics, the number of infecteds never really takes off. And in a lot of cases, you can actually see here that the number of infected individuals actually goes to 0, meaning the disease goes extinct from the population. So the take-home message here, I guess, is that demographic stochasticity can be really important for driving the overall dynamics of an epidemic. And it's especially important when the overall number of individuals in our population is small. So at the very beginning of an epidemic, say when the number of infected individuals is very low, the individual fates or the individual outcomes of unique individuals is very important. It really matters whether a given individual transmits or whether that individual recovers before it has a chance to transmit, because that will determine whether the epidemic is actually able to take off or not. And so we can use the stochastic simulation algorithm to basically simulate any type of Markovian process. Remember, a Markovian process is just any system where the future dynamics only depend on the current state. And so in this case, our SIR type models are a perfect example of a Markovian model, because what happens next depends only on the number of susceptible and infected individuals that we currently have. So we can use this type of stochastic simulation algorithm to really simulate the dynamics of any dynamical system with stochasticity. I've shown the general algorithm here, but it's basically very similar to the three-step process that I just outlined for you for the SIR model, so we won't go into the details again here. I just wanted to note that this algorithm is sometimes referred to as the Gillespie algorithm in the literature, because it was first developed by this guy, Thomas Gillespie, who was interested in the stochastic dynamics of chemical reactions happening when you had a small number of reactants, say a small number of enzymes and other biomolecules within an individual cell, where you can't just assume that there's a large number of reactions and so that the reaction will proceed deterministically. Okay, so before we move on, are there any questions about demographic stochasticity or how we would simulate it under these stochastic simulation algorithms?
Hi, David. I have one question. And the stochastic model is to add the noise to the deterministic model.
Yes.
Oh, okay.
Yeah, so we were just talking about the different types of stochasticity, and really this stochastic simulation algorithm that we've described is really about how we add demographic stochasticity. So randomness or variation due to the sort of discrete nature of our population, of that we have discrete individuals, and those individuals can vary in the times at which particular events occur, say birth and death events.
Thank you.
No problem. Okay, so, well, I'll just ask if there's any other questions. Not hearing any, so I'll move on. The really cool thing about the stochastic simulation algorithm, I think, is that it can really be used to simulate just about anything, not just epidemic dynamics, and not just things in molecular epidemiology, as we'll consider, but really any type of physical, chemical, or biological process. And I think it's also sort of neat to think about, because we're simulating in a way that we actually think the world actually works, right? So there's generally a discrete number of actual individuals in any given population, whether that's the number of sort of chemical molecules in a chemical population, or the number of populations, or the number of individuals in a human population. The world at some level is oftentimes actually discrete, but it's continuous in time, meaning the next event can happen at any point in the future. So by simulating under these stochastic simulation algorithms that allow for continuous time dynamics, but discrete numbers of individuals, we're capturing something about how we think the world actually operates. And so we can use, or we can apply these stochastic simulation algorithms to simulate realistic data, not just for epidemics, but we can also use these algorithms to simulate, for example, phylogenetic trees, which we'll consider next, and also to simulate molecular evolution along particular lineages in a phylogeny, so we can simulate sequence data as well. So first, since we already talked about how we add stochasticity to our epidemic dynamics, we'll talk about how we can use the stochastic simulation algorithm to also simulate phylogenetic trees. And this turns out to be quite easy given what we've already discussed. So in the stochastic simulation algorithm that we just described for simulating an epidemic, there was both transmission events going on and recovery events going on. But when a given reaction occurred, we didn't really consider the identity of individuals. We didn't actually consider who in the population was getting infected or given recovered. But if we want to simulate transmission trees or phylogenetic trees, then the identity of these individuals actually matters, because the genealogical or ancestral relationships amongst these individuals in our tree is going to depend on their individual identities. So we can run the stochastic simulation algorithm to simulate an epidemic, but to get a phylogeny, what we have to do is be able to record exactly who in the population is getting infected by whom over time. So instead of just having a generic reaction, what we'll record when we simulate phylogeny is exactly who is infecting whom and at what time the individual transmission events occur. So we'll record, for example, in this case, that it's Bob that infects Jane, and it's Jane that infects Joe. And we can use these identities of these individuals to then reconstruct the given transmission tree by then looking backwards in time. So to reflect reality, we can consider the fact that generally we won't be able to sample everybody who becomes infected in our population. We'll just sample a given number of individuals, the individuals here that I've highlighted in red. But since we've recorded the sort of parent-child relationships in the transmission tree as we were forward simulating through time, what we can now do is walk backwards through time and trace the ancestry of particular individuals and then get the resulting transmission tree. And what's sort of neat about this is you can do it in either forwards time or backwards time. So I just described this in terms of the forward time algorithm of simulating an epidemic forwards in time, but we could also do it for a backwards time model, say like a coalescent model, where we could just use the stochastic simulation algorithm to simulate when a particular coalescent event occurs in backwards time and simulate a genealogy that way. That's actually the more standard way of doing it in population genetics. But in epidemiology, we're normally interested in the forward time dynamics of the epidemics unfolding. So we generally forward simulate the epidemic and then track the ancestry of particular individuals backwards through time. So it would be pretty easy to code up or program our own stochastic simulation algorithm. It would just take us a few lines of code, but it would become much harder if we also wanted to simulate transmission trees or phylogenetic trees while we're simulating our population dynamics or epidemic dynamics, because we would also have to track the ancestry of individuals backwards through time. So fortunately, there's very efficient software for doing this for us. One nice package is was written by Tim Vaughn, and it's the master package in Beast 2. And what master does is it uses the stochastic simulation algorithm to simulate population dynamics as well as trees forwards in time with a model that includes demographic stochasticity. So we'll get a chance to actually play around with this in the tutorial on Wednesday. For now, I'll just say that it's fairly easy to use because all you have to do sorry, is formulate your model in an XML file, typically like the XML files that we standardly put into Beast. Say or tell Beast what types of populations or what types of individuals we have in our model. and then specify the individual types of events, or what master refers to as reactions in the model. So for example, that an S and an I individual can react to form an I infected individual and then an exposed individual who hasn't yet become infectious, like in a SEIR model. And then we can use master to simulate not just the epidemic dynamics, but also phylogenetic trees, like the tree on the right here, which not only shows the ancestry of the individuals in our population, but also shows when particular events occurred. For example, whenever a blue lineage turns yellow or red here, that means that some exposed individual is becoming infectious. So we'll get to, as I said, play around with master on Wednesday, and it's actually, I think, a great deal of fun because we get to simulate cool trees like this fairly easily. Okay, so we talked about how we can simulate phylogenetic trees. The next thing that we consider is using the stochastic simulation algorithm to also simulate molecular evolution along a given phylogeny, so we can actually generate simulated sequence data. And this is another pretty straightforward extension of the stochastic simulation algorithm. So we'll assume here that we already have a given transmission tree or phylogeny that could be sort of the real tree or a simulated tree. And then we'll assume that sequences evolve along each lineage in this tree according to some model of molecular evolution, like an HKY or a GTR model that we've seen already many times in this class. But then if we think about sequence evolution in this way, simulating sequences along particular lineages in the phylogeny becomes very similar or results in a very similar algorithm to what we've already seen. It's the same basic three-step model. So our first step, if we're simulating sequences along a particular lineage, say this dotted lineage here at the bottom of the tree, is that we have to decide when the next mutation or substitution event occurs. So say we're at time t here in the phylogeny at this branching event, and we know this lineage is currently in state A at a particular site in its genome, then what we have to do is be able to draw the time to the next mutation event. So as before, we can draw this event from an exponential distribution. The waiting time to the next event can be drawn from this exponential distribution with a rates, lambda total. And in this case, the total rates at which mutations or substitutions are occurring will just be equal to the sum of the rates of all the individual substitution events. So for in state A, there's three possible mutation events that can occur. We can go from A to T, A to C, or A to G. So we sum all the possible events together to get the overall substitution or mutation rate. And then we can use that total rate to draw the time to the next mutation event. Then at step 2, what we have to do is, as before, decide what type of event occurs. In this case, we need to decide what type of mutation event occurs, so we can compute the relative probability of each type of substitution occurring. I haven't listed out all of these, but for example, if we want to consider the relative probability of this particular site mutating from A to G in a nucleotide substitution model, we can consider the rates at which mutations happen from A to G over the total rate at which mutations occur to figure out the relative probabilities of each substitution type, and then randomly pick what type of substitution event occurs by drawing a particular event relative to their relative to the probability of each event type occurring. And that's pretty much it, right? So once we know the time of the next mutation and what substitution occurs at that mutation, then we can actually just update our system. We can say that if the mutation is an A to G, we can change our sequence and we can move ahead to the next time at time t plus delta t. So if we repeat this process along each lineage in the phylogeny, always starting from the ancestral sequence or ancestral trait at each branching event in the tree, and we repeat this for each site in the genome of our pathogen, what we can do is we can simulate an entire sequence alignment at the tips of the individuals. And this is actually how most sequence simulation algorithms actually work, using very similar types of algorithms. Again, it would be relatively easy to program or code our own simulation algorithm, but fortunately, we don't actually need to do that because there's plenty of good software out there that already simulates under these simple molecular evolutionary type models. I'm not actually familiar with all of them out there. The ones that I've used in the past that I like, though, are SeqGen, which has been around since probably at least the late 90s, and it's used to simulate molecular evolution, especially for like viruses and bacteria, using simple types of substitution models like HKY and GTR models. And there's actually a hidden implementation of the SeqGen model in Beast 2 if you want to use it. It's just not widely advertised, but this website here, or this URL, gives a link to a web page that describes how you use the SeqGen package in Beast 2. The sequence simulation package that I've started to use a lot in recent years is PyEvolve. And I really like PyEvolve because it's incredibly well documented and easy to use, and it's already in Python, so it works well if you're already working in Python. And it can not only be used to simulate under simple HKY or GTR models, but it's also nice because it can be used to simulate under much more complex scenarios. For example, if you have different genes in the genome of, say, like a large eukaryotic pathogen that are evolving at different rates under different types of substitution models, you can actually split up your genome or your different loci into different, what PyEvolve calls partitions, and then simulate sequence evolution under those different types of models. So PyEvolve is quite nice in that sense. And on Wednesday in our tutorial, we'll actually use PyEvolve to simulate some sequence data. Okay, so we just spent a lot of time talking about how to simulate sequence data and how we can actually do simulation studies where we generate particular phylogenies and sequence data. But why do we actually need to be able to do this? Well, I think ultimately it's to actually serve as a sanity check that our methods and our models are actually working like we think they're working. And so we generally do simulation studies because a number of things can go wrong. There can be errors in the packages or software that we're using. And this is particularly common with scientific software, because scientific software is, at the end of the day, mostly written by people like you and me, graduate students, postdocs, assistant professors, people who weren't necessarily trained as software engineers, and so could easily make mistakes when they're implementing a particular method in code. So running simple simulation studies allows us to check that the software is actually working like it's supposed to. Another reason that we might want to run simulation studies is there might be error at our end of things too, as an end user. So not actually bugs on sort of the development side, but bugs actually in our use of a given software or package. And to convince you that this is a problem, think about, setting up a complex analysis in Beast. Beast is fortunately pretty well documented, but there's lots of other scientific software out there that have similar sophistication or complexity of Beast, but are a lot less poorly well documented. And in those cases, it becomes very easy to make a small mistake as a user, where you say, misinterpret some sort of input that the model expects, and you might get the completely wrong results because, for example, there was an argument or input parameter in the model that expected a rate, and you used a probability instead of a rate. Another reason we might want to do simulation studies is to make sure that we're not generating or adding in errors into our own computational analysis. And the third reason we might want to do simulation studies is to check the statistical validity of the model that we're doing inference or estimation under. So a lot of the programs that we've used in this class, like BEAST, try and estimate particular parameters or try and infer phylogenies. And we would like to be able to make sure that the models that those types of methods assume actually reflect the way that the data was actually generated in reality. But this isn't always the case. Just think about some of the models that we considered in this class. A lot of them assume that we have, say, a haploid population of individual pathogens. And so if you wanted to apply one of those models to, say, a pathogen that was diploid, it might be a good idea to actually check if that model or method is appropriate by doing a small simulation study where you actually simulated sequence data or a phylogenetic tree for a diploid population to see if the assumptions of the inferential model or the statistical model are actually appropriate. So what I recommend, especially in molecular epidemiology, where our analysis tends to be quite complicated because we both have complex data sets and we're generally running complex analyses and things like BEAST, is to do a simple sanity check where we sort of play this game of, can we get back out what we put into our simulations? And so this is also like a three-step process where we would first simulate data under known or what we'll consider to be true parameters. We then use our favorite inference method, say BEAST, to try and estimate the parameters that we've used in our simulation studies back from the simulated data. And then we can compare our estimated parameter values to the quote unquote true parameter values that we used in the simulation to actually make sure we get back out sensible results. We get back out sensible estimates from our simulated data. This is, I often think about, exactly the same as sort of doing controls, positive or negative controls in laboratory science. We don't do positive and negative controls when we're doing experiments in the lab because we think it's fun or scientifically revolutionary, but it serves as a basic sanity check to make sure that the results that we're getting from our experimental protocols aren't complete gibberish or complete nonsense. And being able to do these simple types of simulation experiments when we're doing computational analysis serves as a similar sort of sanity check. So I can tell that I'm getting quite pedantic by the end of this lecture. So I just want to end by saying, I think that performing these simple simulation studies is a great way of being able to identify preventable errors that can easily seep into our analysis, either because there's bugs in the actual scientific software or we've caused an error in our sort of input into these analysis. And that if we did these simple sorts of simulation studies, we would probably make our science much more stronger and much more reproducible in the long run. And so in the tutorial on Wednesday, we'll actually get a chance to do this. We can sort of work through one of these simulation study protocols for molecular epidemiological data. So what we'll do on Wednesday is we'll simulate some pathogen sequence data under an SIR type model. And then next week, what we can do is we can use BEAST to actually be able to check if we can actually estimate the parameters of that SIR model from the epidemiological models that we're using to actually see if BEAST in our inference methods is actually working correctly when we're trying to fit these SIR type models.